Hi, I'm Charlotte Allender and welcome to one of the glasshouses in the UK Vegetable Genebank. The purpose of the Genebank is to conserve genetic diversity for us in vegetable crops. Having access to whole gene pool diversity in crops is really important for plant breeders and researchers in order to understand the genetic processes which control traits of interest that farmers need, but also to make sure that breeders have access to new sources of variation in order to create new varieties to meet the needs of farms in the future. Here at the UK Vegetable Genebank we specialise in vegetable crops, but in the UK there are several other gene banks that focus on cereal, on forage crops and on fruit trees. So here in the Genebank we keep our seed under long-term storage conditions. So for us this is minus 20 degrees C, but to be stored at that temperature the seed needs to be really dry. So all of our seed samples are dried down to around 5% moisture content by weight. And we keep the seed under those conditions because it really reduces the metabolic activity of the seed and enables the seed lifespan to be extended as long as possible. So some of the seed in our collections could last for decades, if not longer. We're still holding, and it's still perfectly viable and usable, seed that was donated to us in the early 1980s and is possibly actually even older than that. When seed is stored at minus 20, as I say, it remains metabolically active, but at a very low level. So we need to check the health of the seed samples, and we do that generally every 10 years. We do that by germination tests. We will take out a small sample of the seed stored and test the viability by germinating it on filter paper in an incubator and counting how many seeds germinate. And we'll compare those readings between tests to see what the health of the seed is doing, and if the germination level drops below a set threshold, then we will actually shortlist that seed sample for a process called regeneration. We select the samples we need to regenerate from based on a number of priorities like the germination levels and also expressions of interest in samples that are otherwise not available because of low seed levels. We will sow those out. So for example, we will sow our brassica samples out in July. We will sow our carrot samples out in September. We will grow the young plants on to a stage in which we can pot them on and place them in our isolation cages. So each seed sample will go into its own isolation cage, and we do this because a lot of our material is outcrossing, so it will readily cross with other plants of a similar crop. And we want to avoid this because we're trying to maintain not only diversity but the integrity of each sample in our seed collection. So we use the isolation cages to keep the plants and the pollinators we use separate. So we use blowflies as pollinators. They're not particularly attractive insects, but they are very effective. Quite often we will have our isolation cages with lots of plants in and a fair number of flies buzzing around doing the pollinating. So the cross-pollination enables us to get a good seed set. So many crops like brassica and carrots are outcrossers. The carrots in particular suffer from something called inbreeding depression. So we need to transfer pollen between as many plants as possible. We generally start with 50 plants per accession. Each of our seed samples you can think of like a population, and so it's important to retain the genetic diversity present in that population into the next generation, and that's why we use 50 plants and not say 10. We harvest seed from our brassica crops in May and June once the seeds have set and the pods have dried back. So we will go through our isolation cages, snipping off pods and placing them into paper bags. We'll do that process for crops like carrot later in the year in September. Once harvested, the seed heads and pods in their paper bags or their trays will go into one of our drying rooms to dry off, and this just makes the plant material nice and brittle, so it's easy to remove the seeds from it and clean the seeds out. This seed cleaning is a really necessary process. We also have to be very careful because plant dust and debris is actually hazardous to health in terms of being an allergen. Once it's cleaned, we will dry the seed back some more. Once the seed has been in the drying room for at least 2 weeks, quite often longer, we will check the moisture content of the seed using a hygrometer. We place the seed sample in a container and actually measure the humidity of the air just above the seed sample. And this gives us an idea of the moisture content of the seed. So we are looking for something that is equivalent to 5% moisture content by weight in order for us to be able to package and store the seed. And we will weigh it and carry out a germination test to get an initial idea of the health of that newly regenerated seed. And then we will package it into foil pouches. These foil pouches are really important for long-term storage because they will prevent the dry seed from reabsorbing moisture while it's in the -20 store. In that way, it will prolong the lifespan even further. With a collection of 14,000 seed samples, it's really important to actually manage the data associated with those seed samples. And this is really important in order to keep track of our stock levels, so how much seed we have and actually where it is in our -20 stores. But we also need to manage the data about the provenance of our seed samples. We call this passport data, and this describes the species, the crop, where it was collected from, and any environmental and other information associated with that seed sample. And that can be really important to help users select what material from our collection they would like to use. Our material is used in quite a range of research projects, everything from looking at crop evolution and domestication right through to looking for specific traits that are of interest to plant breeders and their customers, the farmers and growers. I hope you've enjoyed this virtual tour of the UK Vegetable Genebank. If you'd like to find out more about our work, our collections, or even how to request seed from us, do visit our website. All the information you need is there.